Homology-based inference is the topic today from two different perspectives. Let's begin with the perspective that we began on last Thursday. There we started talking about the prediction of subset localization. And today we will begin with the question of enzymes. So one way of calling this question is, how well is enzymatic activity conserved through evolution? Now, this was Dimitri. Thank you, Simon. Yeah. Oh, there was a different rationale. I thought that Simon wanted to open the door for a newcomer. The other question essentially is the one that we are after here in this context. This is not a good place when the next one comes in. Can we predict enzymatic activity through sequence similarity? Can we predict at least that an enzyme that the protein is an enzyme? And the first question that I have to you, so I showed you on Thursday how well we can do homology-based inference for subset localization. So for the question, how easy is it to infer that one protein is nuclear if it is similar to another protein that I know is nuclear? So I'm going to ask the same question here for enzymes. My question to you here is, what do you believe is easier? Or what do you believe, where will you be more accurate in your guesses? Thank you. I'm sorry, I started early because of some expected changes of the routine today because of an unregistered visitor that we have, who will be with us most likely for several meetings. So the quick guess, the question is, when we look at, and again, I'm sorry that I started early, the regular time is 12:15, so you're all still early in time. We want to infer, yeah, that's the problem. No. So this is the visitor you may not have seen yet. Unless she falls asleep again, you may all get to Nora. So I can infer that a protein is nuclear from the fact that it is similar in sequence to a protein that I know is nuclear. And I can do the same thing for enzymatic activity. For instance, I can infer that something is a transferase. What do you believe works better? I understand that you have no knowledge of the answer. It's a pure guess, and your guess is as good as any. Can you guess, and can you argue why you guess that way?
I think an enzyme, because the structure is related with the function of the enzyme, and with a protein it's not necessary.
With the subset localization, it's not necessary. That's a very great point. That's for me, would be a very, very good reason to formulate an expectation, and everything you say is right. So we would expect that we will get back in a little bit more detail about this in the context of subset localization. But in principle, enzymatic activity is coined by a particular binding site, histidine triad, when we need three histidines next to each other in a particular spacing in three-dimensional. So it's clearly related to the structure. So this is clearly something that you expect to be more similar. I don't have a photo of you.
I could be. Yeah.
Right, and neither a photo nor a name. I realize now. So can you please also send in the photo to assistant at Rost Lab within?
Yeah.
Thanks. Does anybody have any other guess? Opposing or a different reason for the same guess? So the guess is enzymatic activity will be conserved more, will be easier to infer by homology-based inference because it's more related to structure, because it is a particular binding site that is coined by the structure. Anybody has a problem with her roaming around, tell me. No other guess? Okay, let's get back to the description of enzymes. Enzymatic classification, or enzyme classification, EC, so-called EC number. The highest level of the hierarchy has 6 different classes: oxyreductases, transferases, hydrolases, isomerases, ligases. And I missed the liases here. And then in the second figure, so the second digit of a 4-digit number, is slightly different in the different groups here. Here for transferases, they transfer a particular molecule or the group, and the second level is distinguishing the type of group that is transferred, and so forth, and so on. This is a strict hierarchy, meaning that the further you go down, the higher the level of detail. Also meaning that if you have a fourth level, two fourth level annotations are comparable with each other in the density. So whether it's 4.1.3 or 2.1.3, both have a similar density of information. That is not the case for Go. So in Go we cannot compare completely different classes, even within molecular functions. So even within one Go category we cannot do that. For enzymatic classification we can. We typically assume that 4.1.3 really is much more similar to 4.1.1 than it is to 3.1.3 or something like that. And largely that is true, although on that level of the detail of the chemistry, sometimes essentially the way the bond is treated is similar to between a transferase and a lyase, is similar across these categories. So one aspect of the reaction is similar, but overall we believe it's we treat it as a strict hierarchy. Now here comes the first dent. So not only do I not have a photo of you, what is your name?
Neil.
Neil. I-A-L?
N-I-E-L.
Neil. Ah, Neil, like German, like Swedish or something.
No, Belgian.
Oh, Belgium, okay. Now comes the first dent in the image of Neil's idea. It is true that the detail of the structure really matter for the enzymatic activity. But here we in fact have an example where we have the same enzymatic binding site, the same enzymatic reaction catalyzed by very different structures, very different positions in the structure. So yes, we need a scaffold. And yes, this scaffold essentially is always doing that enzymatic activity, and that scaffold is doing that. But they actually have evolved the capability of doing something very similar with very different scaffolds. Now, if, again, we want to infer the homology-based inference, or well, in homology-based inference it does work, we take all proteins, find the subset of all enzymes in all of these proteins, take all the enzymes of known function. That's essentially what we have. And now we do an all against all, right? All against all means we pick out everyone from this data set and we pretend we don't know the enzymatic activity it has. And we simply say, can we infer by finding the one that is most similar to it in terms of sequence, can we correctly predict its enzymatic activity? Right? That's homology-based inference. And here's one answer to that on this side here. That side here relates to structure. Let's not go there for the time being. We have identity classes, so pairs of proteins that are 95 and more, 98, 85, 75, and so forth. Blue means that all 4 digits of the enzymatic activity are conserved, are identical. The purple coming up here is that the first 3 are identical. The fourth is no longer. The next one that comes up is the gray here. That means 2 and 1. 0 essentially means still it's an enzyme. Okay? No, actually 0 means 0. 0 doesn't mean enzyme. Because all of the, so we are not measuring enzyme versus non-enzyme because all of those here are enzymes. So the purple is entirely wrong in terms of enzymatic activity. We have 2 enzymes, we have a pair of enzymes that are aligned, but they don't have the same number. So the conclusion from that is, does it work? Can we infer by homology enzymatic activity? Let's begin with your no. Why no? What is it that you don't like? Not everything is 100%. Is that the no part of the?
No, I was thinking about sequence. What are those identity
Okay,
classes?
so let me go back to that. Identity classes means my data set is not big enough so that I can plot every single integer, for instance, right? So I have to bin it, okay? And this is what this word means here. We bin all the proteins that have 95 and more. all the ones that are between 85 and 95, all the ones that are between 75, and so forth, and so forth, and so forth. Yeah, that's most likely what I see. Everybody clear about what the graph shows? My question to you, does it imply that we can homology-based infer enzymatic activity? No.
How do you calculate the identity class for identity for enzymatic function?
Okay, let's step back. What we do is we build a database of proteins. In this particular case, I have 5 proteins. Each of these 5 proteins, I know the EC number for. Let's call that the data set of known enzymatic activity. So every one here has an experimental annotation about the EC number. So now I take a protein of unknown and I line it against all of these 5. So through the database, right? And I can use a variety of scales to determine that U and any of these K, 1, 2, 3, 4, 5 has a similarity. The simplest way in which I can sort of measure the relation is by percentage sequence identity. So for every one of these, I can measure the number of residues that are identical, right? And what we have here is all the pairs. So now I want to see how well homology-based inference does. In order to see that, I ask if U has 95% sequence identity, does it have the same annotation? So that sounds bad, right? There was no hard object that I remember on the ground. Well, okay. Anyway, so these would be all the pairs that have not. What do you have in there? What is that? Now show me. Is that a? I don't know. It's amazing what babies can do that is not good. So this is an average of a sequence identity. All the pairs that have 95% sequence identity, again, 100% of those have 100% identity. I mean, all 4 EC numbers. What do you have in there, Jerry? Can you turn around this one? Whatever. Are we now clear about what it means? My question again. Does it imply that enzymatic activity is conserved? Does it imply that we can do homology-based inference? Who says no and who says yes? Who says anything and please argue for it. So now you lie this way around and I can see what it is, huh? Yeah, you don't want me to have that. What is that? A stone. Yeah, okay.
I start just because I didn't forget. The 0, 1, 2, 3, 4, is it?
So what this one means is 4 EC digits are the same.
4. So it's all levels.
Exactly.
The same. Okay.
That's what it means. 3 means 3 of the 4 are identical. The 4th is not. That's purple. Gray is 2. The first 2 are identical. The last 2 not. And when I say 3 identical, of course I mean the first 3. Not any 3. The first 3 identical. First 4, all 4. First 3, first 2, the first digit is identical, and no digit is purple. Here. Now, can anybody argue? Are you? Yeah.
Yeah. The more identical they are, the more likely they have the same function.
Yeah. It's the same enzymatic activity. So, but overall, would you argue that they essentially that is very, very true? Is a correct observation, important observation. Would you argue that it's high?
Well, even at a quite low sequence identity at 35%, most of the proteins have 1 or only have 3 or 4 same terms. So
65 or more than 65, at even 35, 3 almost to 97%. So it's a tiny, even at 35%, you have a tiny fraction that are not at all the same enzyme, right? That's a fairly high number, isn't it? It's certainly, if you remember, I don't know whether you remember last week's Thursday, but it's certainly much, much, much higher than subset localization. Now, this is the way we did it, okay? Is there any dent in the picture? Okay, ideally we would want to do it for all enzymes, but we don't know all enzymes, so we cannot do that. Yes, there are some mistakes, but some unavoidable mistakes. But is there something that you find is weird that you could possibly avoid? Well, we're looking at enzymes only, so excluding the green. One issue that has not been considered here, but given the numbers we saw, this shouldn't matter, is whether there's a confusion between green and blue. Because we are only looking at a subset of enzymes, we would never see that confusion, right? We never really push green ones into the fold, and maybe that would make a difference. But again, the levels are so high, as you correctly observe. All are wrong is only for, at 35% sequence identity, only for a few percentage points. It's a tiny number. So we would not necessarily assume that this matters. We should check this. This is one sort of dent in the picture. But there's a more serious one in here. And that one has to do with bias. Let me go into something else. And it's the issue that we in fact do an all against all. So if that set would have been biased by particular enzyme classes, for which we have many, 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 many experimental copies, then we would toss those in again and again and would count them many times, because we're actually looking at the square effect. So any bias is squared because we're looking I against all. So it's essentially by looking all against all, we square the number. So if we have n enzymes, we do actually n square minus 1 divided by 2. But essentially it's something in the order of n square half comparisons, right? So if there is bias, we square the comparison divided by half. But essentially we have that. So we should in principle do something like that. We should start with a unique subset and compare that against everything. And that brings me to the insert of Zipf. Does anybody know him? George Kingsley Zipf is an American born who actually studied in Bonn and Berlin. Known for, not surprisingly, Zipf law. Zipf law established originally for word frequency. So he discovered that when you look at different languages, they have different words that are most commonly used. And that in fact, even in the English language alone, when you look at the word frequency, so this is the frequency of the word that is ranked n. So n equals 1 is the top ranked, 10th top ranked word, is essentially 1 over the n by a. A is essentially 1. So 1 over 10, 1 over 1, right? That's the function. The function looks like this in principle. Now, the remarkable thing, so that described, he figured out in the 30s that this describes the word frequencies in English, and that there's something that is like that also for other languages, for German. That was the next one he tested. And that was sort of the first time going at languages, a linguist really counting words and doing things. By the way, my strange behavior in moving, I tried to nail her to my feet at the moment. Last time it worked, so I'm not moving because this way I keep her sleeping, hopefully. Which is a little bit odd for me. I need movement. Now, here's a different story. Assume you plotted the size distribution of settlements, cities, towns, and everything. You looked over the entire globe, and you asked how many cities do you have with 25 million, how many with 20 million, blah, 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 blah. Right? What kind of distribution do you expect? An exponential distribution. So now we get into the real limits of my position here. I have my... what exactly do you mean by an exponential? Something like this?
Yeah, like basically, it's like just zips within maybe like with more than 1 or about 1 depending on the value?
Actually, the remarkable reality is the zip. Here is, for instance, so you were very close. So here is the story for metropolitan areas in the US. And you see it's an amazing fit to the zip. And people have since done many, many examples of this sort of behavior, not only for settlements, for sizes of bridges, and if you think this problem through, then you will see that whenever you have anything that evolves under competition, any system that evolves with a competition for resources, whatever that is. For the bridge, you simply have stability issues with the length versus the weight of the bridge, or there is a payoff. And once you go beyond something, you have to do something on top. And somehow this is the same here. Whenever you have something that evolves under competition, you can actually simulate in a computer that you would get a zip flow. Something. No, what exactly the degree here is a different story. Now, what will happen if you picked random points from a zip? What will be the distribution that originates from that? So I let you pick any number of points out of this zip distribution. So we take away, for instance, Shanghai or Tokyo. We take away all the biggest cities. Or you can that's systematically taken away. That's unfair. No, it has to be random. I can change. Oh, what happens if I took random points out of the zip distribution? What is the distribution I get? So the answer is you get a zip, just a lower zip. As long as you choose randomly, and I almost gave myself away. If you can of course pick things to change the distribution. But if you randomly pick point, you will a random subset of a zip is always a zip. Now, here we get to the question: if I plot the number of protein families or proteins per family, so how big is a protein family? Okay, and here's the histogram. What do you expect? Will it look like something like this, like a Gaussian, where there is a certain size that is favored? Very few have very, very large families, very few have very, very small families, and there is some Gaussian that is optimal for the evolution. Will you have something that is sort of linear? So we have a lot of small families and fewer very, very large families? Or will we have something that's flat? That the number of family, or the size of family has absolutely no relation. So it's just, it's equally probable that you have a family of 30 as a family of 300. What do you believe? Oh, is there, in this particular case, a quartum datio, so it could be a fourth.
I think the one that is really nature is the one that basically we could figure it out, because I assume that would be exponential, but then I think it's you have to figure out each one of the smaller protein classes, right? I'm not entirely sure what you're saying. Are you saying, I don't know, you have to look? No, is it the distribution as it is, per experiment, or distribution that people expect in the nature? I'm not doing an experiment with you. You're off the hook. I'm not sending you to program it and check out the database. I'm just letting you guess. So what do you guess? Exponential. So what is an exponential? So that means essentially an exponential means none of the above. Is that right? So you expect something like a zip. Anybody else? In fact, that's exactly what I would expect. The zip, again, we have a competition for size in some sense, for similarity, and some fall out and form a new function. And we need a new function because we need different functions in a cell. It's the same sort of balance of the effects. Okay, now let's get back to the enzymes. What I'm going to do now is I take all the proteins of known experimental enzymatic activity, and I simply plot the size of the family. Red. I do an unbiased sampling. An unbiased sampling says I take an unbiased subset and compare everybody in the unbiased. Say I have 10,000 enzymes, and I have 1,000 sequence unique enzymes. I do 1,000 against 10,000. That is blue. While 10,000 against 10,000 is red. What do we like?
Big families are better understood.
That is certainly correct. That is certainly so. There's a predominance if we take the data as is of large families. Large families dominate. They do not dominate because there are more of them, but they dominate because there's an equal number. It's a totally flat distribution. True? Anybody wants to say anything else? So if, for the family size distribution, this is what we expect, and if the red is what we see, then something went wrong, right? If what we expect is a zip, and if what we see here is a zip in the unbiased, then that obviously samples the expected distribution. Now we get back to your question. Is it the distribution that we expect today to be able to measure, or is it the distribution that we expect the database in 100 years to be, right? So the universe of all the families. Today, the best we can do is say, well, what we have today is a proxy. We assume it's a proxy for tomorrow. So we do know today is still biased, and we will see something different. But one thing is clear, that taking the bias at face value is much worse than the blue. Maybe the blue is not the true distribution, but most likely the blue is much closer to the true distribution than the red. So it tells you on an issue that has nothing to do with homology-based inference of enzymatic activity, but simply sort of a cross-check, a sanity check of your data. You see that using using the entire bias creates something that is nonsense. This family size distribution is total nonsense, right? So I'm going in this level of detail just to tell you, you have to sometimes find a way to look at your data in a different way. I just look at my next slide and there's something wrong with it. In a different way, meaning, so sometimes before you actually do bias reduction or not bias reduction, try to find some other reason where you can see which of the two will be right. Sometimes maybe there may be reasons for bias. Here we see using the bias is nonsense. Now, so ignore this one here. That is where my, and I see that the next two slides are getting wrong. So this is percentage sequence identity, and the data is, I did fit, so it's not really true data, it's close to true data. This percentage of proteins. So again, I'm asking, if I do this here, for the entire set of known enzymes, for how many do I find that the first EC number is right? All 4 EC numbers are right. That's light blue. This is like what we saw before. And the conservation here is somehow very similar to what we saw before. It's a little bit lower now. Remember, previously this was still higher. But roughly that's what we saw before. We take the database as is. The difference between this version here and the one we saw before is this is a little bit a larger data set. It's a bit later. But in principle, this confirms exactly what we saw before. Okay, that's the biased version. And here's the unbiased version. Again, what we plot here is percentage sequence identity, percentage of protein pairs that have the same. And you see that in the unbiased version, at some point here, at very, very high levels, 80% and more, there's no difference. But very rapidly, so when we go to levels of 50% sequence identity, and that's a realm in which we very often look at alignments. When we go to 50%, then you see that, let's just look at 4 EC digits. Now we're getting to an estimate at 50% is less than 20 versus at 50% is 95. So that's a big difference, right? This is right. This is wrong. Right estimate. So now this all comes underneath, and that makes those are the real curves here that were published. The green, the red and the orange, simply this is accuracy, the blue curves, the bluish shades, and the other ones are coverage. So at every threshold, say 60%, how many would I find? To just let's look at 2 points here. When we find 50%, if we had the biased universe, we would argue that over 90% are right. At the level where we have the unbiased version, for 50% found, we have less than 15% right. And this really is completely turning the story upside down. We're moving from something that is absolutely clearly conserved to something that is absolutely clearly not conserved, right? So, and all we do, we use the same data. All we do is we pre-filter the data in a way in which we should, in order to get the bias of very large families out. Okay, these are the different curves for different EC numbers that differ slightly. What I show here now is the distance from the HSSP curve. So the blue is the HSSP curve that I showed you before on Thursday. So plus, minus is this curve here. That's percentage sequence identity. Again, different EC classes have slightly different behavior. This is pairwise blast, this is ply blast, this is the E value, this is the distance from the expectation value, and you see that psi blast does better than pairwise blast and that this one does very well. Essentially, that's it. Let's get back to the point that I asked you before. And here is the H value distance, here is the blast E value. Green is the conservation of structure. This is such a sharp transition because the curve here has been optimized to exactly fit the data somehow. Blue is subcellular localization. Red is all 4 EC numbers. Orange is 1 EC digit. And we are back to what Neil said. We essentially have a higher conservation for enzymes than for subcellular localization, which incidentally, if you care to know, for me was not what I expected. So the only person who dared to say something today predicted the outcome. Both red and orange is above blue. And not only that, in some sense, Neil, the reason that you give there is also seen to come through in some sense, because structure is more conserved, right? And so somehow the story, you could argue the story in that sense appears to shake out. Now, essentially what we learned from this is if I want to homology-based infer something, I have to set up a threshold for blue subcellular localization, for red enzymatic activity, and it might be different here, whether I want the level of detail of the enzymatic reaction in red or the orange. The threshold differs. So it's not only that the values differ, but also maybe good cutoffs in the curve are at different points. Okay? Because yes, they're all sigmoids, so the red and the orange are both sigmoids, but I would argue that the orange here is somehow to some point relatively flat. And that here the orange behaves under E value, and the psi blast E value here behaves very differently from the red. So enzymatic activity itself, there's not one answer how well does homology-based modeling work. It is for your tool that you use, for your decision of threshold, what you call sequence similarity, for your alignment tool, psi blast E value, and for your task. Do you want to infer something as an enzyme? Do you want to infer the enzymatic activity on the level of detail of the chemical reaction? And now let's go to a different level. So this is biochemically. Chemical function is structure, localization, enzymatic activity is biochemical function. What if we wanted to infer that two proteins are both members of the same cell cycle? So cell cycle now is a different level of function. Cells divide, and there is a set of proteins that is involved in making cells divide. And in fact, some of these are enzymes. But there are specific enzymes involved in the cell cycle and cell cycle control. So if my goal would be to find all the proteins that are involved in cell cycle by homology-based inference, how would I go about it? That's, by the way, work from Kaz Brzezinski. He's now at the New York Genome Center. When he started working, so cell cycle, there are different phases while the cell grows. We don't distinguish phases here really. We don't want to say that a protein is from the so-called G1 when the cell grows phase or from some cut points. We just want to see is any of the ones that I participate in anything that has to do with the cell cycle. And those are the numbers of experimentally annotated cell cycle control proteins when Kaz started. From Swissport. For all eukaryotes, he had about 600. For yeast, about 90. Okay, so now the question is, how can we blow up this data by using homology? Ultimately, what are the options? We define a threshold like this. 10 minus 3 in the e-value is a good number. I like it, I understand it, I take it. That's one way of doing it, right? Another one is we take subset localization, or we take structure, or we take enzymatic activity, the orange or the red. Which one of these, or you do something else? What would you do? I understand that the answer has to do with the amount of time that you can spend on it, and it has to do with the objective. But say you had time, and say that the database that you produce from this were giving you a publication. So how would you do it? So that somebody cares. So unfortunately the answer is, you have to do the same thing all over again. You have to essentially use those as the known ones, just like we did before, known enzymatic activity. You have to establish how well does homology-based inference work by pretending that you don't know them, by inferring one from the other. And you get again similar curves. Now they have different numbers. It's again the h-val, percentage sequence identity, blast e-value, and again the accuracy drops, coverage increases. At 0, we see that the curve is a little bit lower than, certainly lower than the localization even. So that allows you to now go to the level and say, a particular e-value, I have a certain accuracy, and by the way, first level EC is shown here at that level, fourth level is shown here at that level, so it's actually, so first level is actually higher at that level. But then we go down in the e-value, and we see that enzyme is still conserved. For cell cycle, the numbers drop much more. There is a jump here. But essentially you have to redo the entire thing. And once you have done the entire thing, now you can go and simply run particular organisms such as human and find all the proteins in human that are related to something that is already known. And then at given thresholds, you can define your error rate. So for every entry in your hypothetical cell cycle belonging proteins, you can come with an accuracy that says this is at that percentage sequence identity, at that similarity in terms of sequence, and that gives me an estimate. And this is in fact what he did, and then you can publish a database. Unfortunately, the bad news here is for every new topic that you want to do, you want to talk about structural proteins, you want to talk about anything that has to do with post-translation and modification, you will have to redo that. That's the downside to the story, and there's no shortcut to it. Or can you think about a shortcut? What could you do to escape from this issue? So I call it bad because this is something that is not working on the level of Go. It's specific to some Go annotations, namely, for instance, to the enzymes, not to others. So meaning that essentially you would have to do that, in the most extreme example, you would have to redo that for every Go number, so to speak. And that is obviously not very feasible. But I do not see a shortcut to that. So it's remarkable. So we did a couple of others on this, and we were surprised how much they differed. But let's get into use that what I showed you for something that is much more coarse-grained. Namely the question, how many human proteins have known function? How would you answer that question? Is that a boring question, or is it a complicated question, or is it too trivial? Or so one way is Google and find out whether somebody has written the answer. But let's say that is actually not available. And in fact, there are answers, but let's say I made you do that for an exercise or for a thesis. How would you go about it?
For example, check how many proteins have at least one GO annotation.
So you would in fact go to the Go database, take every single human protein, run it against Go, and ask what fraction has at least one Go annotation. There is a problem in that, and we will get back to that problem over the course of time again. Can anybody spot the problem?
Some of the terms are actually inferred. It's not really experimental.
Yeah.
There's a lot of Go annotations that have the term homology-based inference. So we would have to exclude those.
They all have a flavor, right? They all have a code for how they infer, right?
They all have what is called an evidence code. So the evidence code essentially means what is the evidence for that particular annotation, and that evidence code then would say homology-based inference. True. So you could exclude them. And that is true for Go, and that is true for Swissprot. Yes. Now, does anybody have a better idea? So this one is bought. The problem with that one is, back to my issue now, you still don't have a feel for how accurate would you do that, right? How, so on the one, wait, sorry, back up. On the level of every protein that has a Go annotation in an experimental, every human protein that has an experimental value, that's one. But now, say we had a mouse protein with a Go annotation, and that mouse protein had 95% sequence identity to a human protein. Incidentally, is that a lot? Mouse and human, 95? How similar is mouse to human on a genome?
It's about the same, 95%.
It's very high. So people have been arguing very, very much about the genome. Let's not get into the detail how that is measured, but the numbers that are Googleable and are typically in the press are that mouse and human are very, very, very similar. So 90%, sometimes 80%. But now let's get back to the level of proteins. Is 95% between a human and a mouse protein normal? An exception? So the number is yes. The answer is yes. It is quite an exception. And so the one number, one triplet that I remember is 7077. So what it means is that if I accept that two proteins overlap for 70% of the length, so essentially this one here I would not accept, but something, I don't know, like this I would accept. I would say those two I compare. And if I say that I call them similar, if they have 70% sequence identity, then 70% of the human protein match in mice. Okay? That's the 70-70-70 number. Which means that the idea of 95% sequence identity, in fact, is really an exception. And that this idea that mouse and human have 80 or 90% genomic similarity is, on the level of protein, pure nonsense, actually. Anyway, this is a different story. Now, back again to many proteins between mouse and human, it's very trivial to infer the Go number. So some we would accept to annotate humans, some we wouldn't. So we need a measure of how well do we do, and one way in which we could do that is we could essentially assume every protein is an enzyme. And essentially, when I say I want to understand how many proteins in human are annotated, I mean at least now one digit is a transferase. Okay, that doesn't really give me the function, but it gives me something about that protein. And that defines then this threshold that I showed you, define one level here. Say I take the threshold for 10% error. And I take another threshold here for 10% right. So this is 90% error and 10% error, right? And now I need something, I do something that is similar to what you proposed. Instead of Go, then I took Swissprot. I said every match to Swissprot is a match of entirely knowing the function. Completely not true, but let's assume we do that. And now we take these two thresholds. So for 90% right, we would have about 20% matching in human. For 90% wrong, we would match 50%. Okay? Now this is not, it's a while ago that we did this, 11 years ago, so the numbers are a little bit higher today, but it provides you some ballpark. which is way, way away from what people typically assume. And this is an overstatement. So this is clearly an overstatement. 90% wrong is nonsense. But even that one here, when I say 10% errors, I have a number around about 20%. Again, this assumes that every record in Swisspot is completely explaining the function. So if I gave you enough time to read this here, this is the Swisspot record here starts here, not in the bullet. You see that, in fact, this really doesn't annotate function at all. That's the first problem. The second problem is that most proteins are multi-domain proteins. In most annotations in Swisspot, there is one annotation. Very often is valid for one of the domains. That means, so 20% of the human proteins would have in one domain an annotation. Since most of them have, so more proteins have 3 domains than one domain, so a lot of function is missing, right? So even that 20% then would be an overestimate. And that gets us to values that maybe 10 to 25% of humans annotated. There are 4 major problems of inference of homology. One is obviously the paralogy problem. So if they are similar in sequence, but it's a paralog that evolved to having a different function. You cannot see that, not easily. There are databases that try to distinguish paralogs from orthologs and try to address this. The moonlighting, proteins have some variant of the protein in a different organism may have 2 different functions, and you may not know whether yours has 2 or not. One problem is the domain problem again, so the annotation is for that. Your query overlaps with the template in a region that is not annotated, so you infer wrong. And the similar thing here is a misannotation in the database that could originate in a mistake like that, and could originate by just a mistake in the database. Mistake in the database could be as simple as you did not see that the annotation was not an experimental evidence, for instance. That is not a mistake in the database, it's a mistake in you using the database. And in fact, it's a mistake that everybody in my group has done at least once, including, and I have done many, many more times. Now, how can you go around all of that? Typically, at the end of the day, the only way to go around all of this is you look at the multiple sequence alignment and you ask, what if I believe this is the enzymatic activity that my protein should have? Then there must be histidine triad, for instance, that is conserved. In the alignment, is that histidine triad conserved? So I will argue with details in the alignment. But this is one-by-one sequence analysis. And again, so some people are very good at that, but most of us would take at least a week staring, not doing anything other than this and running a bunch of methods before we can be sure that this in fact really is the function. And in various projects that we did in our group, we saw that using a lot of sequences, using multiple alignments and using structures, we can do much more than using only one of those, but we may still be wrong. And in fact, so let's call it sequence analysis, is something that you may call an art. Okay, so now we get into the Kappa story, Kappa 1, and maybe I'm going to get through a couple of Kappas today. I'm not entirely sure how fast I'm going to go through this. The slides here, thanks to Tobias, who did his PhD here at TUM, and in fact was a BIM master student, who was very, very good at machine learning, had a couple of publications at the moment. He did his PhD, and then essentially left for Illumina in Cambridge, England. And that is back to meta student, and that is back to something that you guys do. So let me begin to skip here. Let me skip all of these. Why? I always have the wrong slide in the wrong place. So it all began. I'm sorry for being out of sync on my slides. It all began with a talk that I gave in a lecture like this, in which I said, essentially, the goal that you have for Kappa 1, homology-based inference, means you build a data set of proteins with known function, you run a sequence alignment, and you simply identify the one that has the highest level of similarity and read off that number. So this is go 1, 2, 3, 4, 5.
That's the highest similarity. So you call your protein U to be Go5. Okay? That was what I described. And this is the way I would write a method section of any publication I do. And then the task was, in fact, to we had 16 students in the group. It was just the same course here. We split them into 3 groups, and each implemented the simple idea. Right? So the background is clear. Similar proteins have similar function. We used Go, biological process, and cellular no, it's not cellular component, only Go, biological process, and molecular function. Again, finding the one that is similar in the database. Here's what group number 1 did. So they had a weighted 6 nearest neighbor. So essentially, okay, my database is much larger than that. You take the top 6 and you do a nearest neighbor weight. Now, whether you have hit, the word hit here means you have defined some threshold in sequence identity or p-value or e-value, and say if the 2 proteins are more similar than that, I call it a hit. And then whether my current search finds 50 in the database, 500 or 5 is a difference. Yes, ultimately I'm going to average over 5 or 6, but how many are underneath, right? This should matter, and that's what they consider here. They have 3 terms per ontology, essentially 3 different models, and each comes with a probability score. Essentially they put, for all the top 6 blast hits, they put them onto the different trees, and then they average over the tree, the hit they find. Okay? Here's number 2. Group number 2. They do weighted k nearest neighbor. So before, no difference here except k before was 6. They have a slightly different continuous scoring scheme. So it's not hit equals above threshold. Here we have a continuous score. Right? And then they have to normalize the score. And then they have some parameter optimization step in here. Group number, so this is essentially the raw template score, then there's a quality score that goes onto the tree, and that's exactly where they optimize the parameters. 3 is again weighted e-value based on nearest neighbor. Again, not a binary cutoff hit, but a real based value score, and a continuous term score, which again is essentially similar to the other one. Now, one issue that we have is the question, how are we going to call something right? Red would be the leaf in this tree here that are experimentally annotated. So red are the points where I know experimentally the Go number, right? Green are the ones I predict. So how right are we? And so CAFA, in my view, still has not really addressed the issue how to best give the answer to that question. It's not trivial. There are many ways of doing that, obviously, and there are many arguments for many of these ways. The most extreme would be to say, well, we only go to the lowest level, highest level of detail, and we ask, is that reached or not? Okay, then we can sort of get a 50% probability if the leaves above it are hit. Both by the prediction, that is good, but still ultimately only a measure whether it gets the detail or not, to which degree it does it. And this is not what so you're currently working on CAFA 3. This is still not what CAFA 3 will do. I believe this is what we should do, but you immediately see the problem with that is that many cases we will not have predictions at that level of detail. So many of our targets will fall out because we simply don't reach, in the homology-based inference, we don't have anything down there, right? We cannot homology-based infer, or if we don't have any cases in the experimental database, if we have truly new ones. The argument behind this overlap, is somehow by going up in the tree and by getting leaves up in the tree, right, you should by yourself, you should score. So methods that somehow reach at least many points up here and hit the right branch, hit the right part of the function, they should get a reward for that, right? So it's very easy to argue for giving scores that score
different parts of that tree. Now, one problem is that the more you score in between, the more weight will come somehow implicitly on the highest level, on saying this is a transferase. So this is the least accurate one, but the one that you most likely get right, just randomly, right? If you just threw random darts. Down here to the real alcohol dehydrogenase type 1, that is very unlikely to hit. But that it's an enzyme is very likely to hit because, I don't know, 30 or 40% of the proteins are enzymes, right? So by somehow weighing this tree, there's a tendency to sort of implicitly upweight very coarse-grained functional terms. That's one problem. Here's another problem. Say we get a particular score, and again here this is translatable to recall and precision. So terms that I cut my tree at different levels. At different levels I have a different recall versus precision point. So each cut in the tree corresponds to one point here, right? Now on top I may have 3 different models, meaning 3 different outputs. So I say, okay, top 6 average this way is the top model, average that way is the second and the third, so I have 3. So if somebody provides 3 models, how can I handle that as somebody who wants to evaluate, assess at CAFA? One way would be, I average. If somebody only predicts one score and that score is really right, the top will count. If they try to put as many as possible, somehow the average will be determining how well you do. The next issue is, we could in principle go for the area under the curve. But now a developer may say, well, my method is optimized to get a very high precision. I want to get right predictions for very few proteins. Because I have an experimental colleague who will work on that, and they will pay a lot of money for the experiments. And at least I want to be sure that if they go for 2 proteins, that one of them will be right by prediction, then they will like me. Okay? So my method really is high here. About the rest of it, I really don't care. I don't care to find all the enzymes. I just care that what I give them to do the experiments on has a high probability of being right. And both of those have equal value, right? Before, there's no generic way of using function prediction method. We cannot say this is more right than the other. How can we account for that? So that means we would pick one point, but which one? Well, one way of accounting for that would be that we tell the developer, listen, you have to choose your threshold. You give me the point that you want us to assess you by. Right? That's one way of doing it. But then they may argue, well, you know, that depends a little bit on how expensive the experiment is. So I give you a range, and in my typical environment, I work with an experimental biologist, and we have a communication going, and then we have a sorting of the scores, and according to what the money is, they will decide 2 or 5 proteins they will put into their pipeline. And for me, the top has no difference from the 5th in that sense. It's a higher probability. But there is no real cutoff that I can provide you. So how could we do that to be, say you all develop different prediction methods with different backgrounds, how could I be equally fair to all of you? Any idea? What would be the fairest thing you can do to any developer? Why don't you do what is the best for whatever method you use, right? So here, so the F score is simple, is one trick to pick a particular point. It's a harmonic average between the precision and the recall and the precision. And now I simply pick the max F1, F score. F1 is the max of all the F scores. So the black is the average over the 3 models. If somebody had only given one model, it would be whatever that model is. And I simply pick the point that gives the max F of all the points. So if you have a method that is really strong here, the max will be there. If you have a method that is strong here, max will be here. If your method is equally good, it shouldn't matter, and you should somehow find throughout a relatively. So this is not quite. So the advantage of a method that is good throughout is not quite measured because you're only looking at one point. Okay, that is a problem in that. What's the criticism of that? Well, it could be that the max F score is somewhere down in just a little bit of a random kind of. So if everything, so it sort of levels out predictions. Is one of the problems. It could be at a higher F score, so that you really have a method that is very good, but the harmonic mean works out such that almost random actually gives a better F score. That would be nonsense. It is also nonsense in the sense that you make a decision on what is right after you know what is right. In principle, you should be able to, before you know what is right, before CAFA has the assessment phase, evaluation phase, developers should somehow be able to say something about probability of their answer, and that should go in here. So that is the method most friendly, but maybe it's too friendly. Okay, so then we did some, in our group we did some random prediction. We had 3 different groups. By the way, let me go back. I described the problem here, homology-based inference. I explained to you how the groups did it. Is that the same? Remember that I told you about the 3 different groups, how they had implemented. Maybe I should have a slide here that flashes the 3 methods again. Do you remember we're going through it? So my observation was, what I wanted was much simpler. I actually wanted them to sort of give one top prediction and see how well it does. They came with much more elaborate means. They had an average, a k nearest neighbor average. They had continuous score averages. They averaged the score over the tree. So they put a lot of intelligence in there, which was not the way I called my method section. So in some sense I felt that they did not implement my method. They did it more cleverly. This is not what I mean. The next issue is that there were 3 different interpretations of my algorithm. So I spent more time on defining my algorithm than now. I spent essentially an entire lecture defining the goal. But they had 3 different understandings. The next issue was that I, looking at their presentations, could not tell which one would be best. So I assumed that the details wouldn't matter and that they would all look the same. And that they didn't. Absolutely not. So there are 2 graphs here. This is all targets, this is only common targets. In this particular case they don't differ. This is random background. So all methods are clearly better than random background. The brown here we get to later. The 3 different methods are green, purple and this blue here. And you see they differ. Not only do they differ grossly in even, so there's almost no overlap. So the blue one here spans this part of the rock curve, while the sort of purple one spans that part. Very different. And the green has only 2 points. Those 2 points are above everybody else. Right? When I looked up what method was best, again, I was surprised. I could not read the method first and then predict where they would fall. First of all, anyway, I had predicted that they would essentially be the same. But even when I knew that this prediction was wrong, I could not go and tell in advance which detail would have been better than the other. By the way, the brown here is a fit through the 3, and that fit you cannot do on the level of the output anymore, so there's a simple machine learning algorithm underneath here. So simple fits such as take the max, average over the values, all of these failed. And that ultimately is in this particular case the case because they're essentially orthogonal, or very, very different. They span very different areas in the rock curve, implying that they sort of capture very different effects, and averaging over the probability somehow ended up getting the worst of all possible worlds. Slide machine learning never got to the green, but at least got something that is above these 2 methods and sort of got, in some sense, spans something that spans the entire graph here and did fairly well.
One clear result was that nearest neighbor approach was surprisingly good, and that technical details matter. And then we got to the Kappa background. So the issue of Kappa, simply the question was how well this function predicted. And it started with Predrag, Radivoljak, Ido Friedberg, and Sean Mooney. Those three started this entire Kappa activity. They found a bunch of organizers, agreed on target functions, agreed on using Go. In the first Kappa, they used biological process and molecular function, not compartment. Go has many problems. One is that on different levels, there's a different level of detail. The Go numbers are the same. We would call them all the same. Whether something is regulate or something is negatively regulate is a different level of detail. And on the Go number, you cannot distinguish one from the other. There is different sort of parent-child relation rules. So this one is more detailed than that. It's just part of, this to predict something like that is less informative than something like that. That's not even easy to do. The next issue here is the different evidence scores. So one particular evidence score in Go annotation, we discussed that before, is ISO, inferred from sequence orthology. And there are different levels here. You can sort of see that already. EXP in Go is inferred from experiment, IDA inferred from direct assay, which actually is in principle the same. Should in principle be the same. And you begin to see that in this evidence score there's a lot of layers of complication. Ultimately, the world should be simple and it should be a clear-cut experiment. But today, experimental biology is no longer like that. Most of those are most of the annotations in Go now are in fact relatively indirect annotations. Now you could argue we take only the subset where we have very direct annotations, right? That's a decision that so far has not been taken. You could say that you completely exclude that, and that I believe is a decision that they tried to take but also didn't take. I'll get back to that in a minute. So there are different qualifiers, they mean something else, there are different Go versions. So when you assess the question, Go versions, Go databases, Swissprot, Go, in principle Go is annotated from Swissprot, and the fact that this is not entirely contained in each other already tells you that the story underneath is more complicated. So we agreed on a Go version, we agreed on a particular database Swissprot, we agreed on essentially thereby on a subset of known annotations. Now, the moment this agreement is made, things are added to the database, things are added to the literature, and some people may pick up these new things. So even that has some issues, and we did run into some issues. Then we need the evaluation measure. We need to essentially, in Kappa we have a prediction phase. That's the phase from now, or from about 3 weeks ago until shortly after Christmas, right? Or New Year? Do you remember what they did?
January.
What? Which?
The 9th of January.
9th of January. Then there's the annotation phase. So that's the phase essentially when they wait for experimental evidence to come in. And then there's the evaluation phase. That's the phase in which essentially the people who look at the prediction can assess how well methods do. So let me just go in Kappa 1. Things have changed slightly, but not that much. When you look at molecular function, so that's enzymatic activity, biological process, which essentially is the pathway, and you ask the new annotation that was added in the annotation phase, how similar is it to an annotation that was there before? And you see that mostly you are very similar. That most annotations that are added, or that we used here, or that the Kappa used, were similar to annotations that were made before, right? So mostly, in some sense, we are in the homology-based realm here. Then we have the issue with the evaluation and the max F1 score, and then we have to annotate. Most Kappa 1, most methods, all methods used alignment information somehow. Some methods also use literature, text mining. Some use interactions. And this is the first 34 scores. Recall precision. You clearly see some things are really bad, some things are really good, and it may not be quite clear what is the best. So this has some strength, this has some strength. When we sort them, molecular function, biological process. So this is just the meta predictor that uses only sequence similarity. It somehow was in the top 10. Here, I believe it's underneath 1, 2, 3. It's also, it's not in the top 10 quite there for molecular function, which is strange. We expect it to be higher there than here. And essentially, what we found out in CAFA is that somehow sequence prediction works very well, but there's room for improvement. Let me go to the tail end here. So the top teams are shown here, and then the bottom teams are shown. This is not a bottom team. That's naive. Naive essentially means you predict according to the probability of that term being in the database. So when you predict this to be a kinase, you simply measure how many kinases do you have. So what's the random odds of getting that? Well, this one here is what I had proposed in my lecture to do. You run a simple blast. Well, I had proposed a psi blast, so it's a little bit a step up from there. But a simple blast, take the first hit. Okay? Is there anything that jumps at you? In the results? No? Does nobody wonder why naive is on the same ballpark or even higher than blast? What does it mean? Well, typically when you observe, this is totally meaningless. You randomly throw a dot and predict a kinase, or you find anything that is a kinase that is similar to something that really is a kinase. So either this one went totally wrong, and the database that you used was not of experimentally validated kinases, but of homology-based inferred or whatever. There were mistakes. So that's, I would call, a programming mistake. Right? That didn't happen here. So what else could go wrong? Ultimately, it turns out that the value that, or the way the methods are assessed is wrong. It cannot be that a random dot is better than blast. If that comes up, then the way you measure performance is wrong. This is nonsense. And the reason why this happened is ultimately that it gave too much weight to essentially low detail annotations such as it's a transferase. Or not even that. Initially, so in the first round, a protein binds. The annotation, a protein binds, was dominating the good performance. And protein binds is a total nonsense, right? Somebody could, in fact, we never had this idea. So instead of calling naive, we could have called it an idiot and predicted simply the ones that would come up in the scoring system. So we could have sort of foreseen that the scoring system goes wrong, and could have developed a method that gets a very high score in that scoring system. And that would have been way higher than naive. So binding, a thing that always predicts binding, is totally nonsense in terms of function prediction. It's really completely defying the idea of CAFA, but would have scored very high. And now, how can you avoid to do that? It is clearly a mistake. How can you avoid making that mistake? Any idea?
We can give a higher weight to predictions that are in a higher and a more precise category.
Can you, that is true. Can you foresee what will happen if you will do that? So how much higher? So you have to come, so, and in fact I'm dwelling on this for so long because it's such an intrinsically bioinformatic problem. So we begin with something very simple, homology-based inference, such a simple concept. Just, run your darn alignment, build your database, run your darn alignment and find the best one. But what do you call best? Best how many? Best over what region? So you immediately face the optimization of parameters that you don't quite know how to optimize. And here you bring in a new one in some sense. In fact, Predrag tried that. So optimizing the parameters, one way of optimizing the parameters is by random background. So the weight is proportional to how often you see a score. So protein binding is 50%, so it's completely down-weighted. something that is much rarer is completely upweighted, right? So you get essentially the data to define your weights. That's the idea there. But in this particular case, you immediately see that the next question, the moment you say weight, completely intuitive, completely right, but the problem next, how to choose your weights, let the data define your weights. Now we got the problem of bias in the database. So letting the data decide means that you choose the data from today. Now which one? The biased version or the unbiased version? Back to the enzyme story, right? And ultimately, so far we have not found a way to do this right. Okay? The way that Tobias has defended, and I believe I'm with him, is to really exclude more of the higher level and just ignore it. What we really care for is the detail. Or another level that Predrag and I have been discussing for a while now is just throw the entire Go number away and think about it as an abstract. So protein function is described in abstract, and what we should actually have is a method that writes an abstract. And then let's just text mine the similarity of two abstracts. So that's a wild concept that Predrag and I have discussed walking, but we haven't sat down and have not really. And you thinking about it, you will immediately find many ways in which this can go totally wrong. Maybe there are more ways in which this can go wrong than in which can it go right. But this is something that I believe we should really do. Okay, so after the CAFA we optimized, we sort of added new targets, and one of the issues then we got a meta student, and the meta student performed after CASP relatively well. In CAFA 1 we sort of redid it, and that's exactly what you do now. You do the same thing, or I believe some of you in the exercises at least redo the same thing with the new terms that are available. By the way, there was something here that is not on those slides, and I believe I would call that the last slide for today. So the conclusion, by the way, from CAFA 2 is that overall this is naive for molecular function, and this is BLAST. So this original problem is solved now. This is the top performers. So the top performers are better. Now the top performers in CAFA 2 was 120 methods. So this is the top whatever, 3, 6, 10 of 120. And you see that the top are all better than simple BLAST, but not that much, right? Yes, for biological process, we have again this naive versus, so there we have a clear case, we still haven't solved this issue. So this is after the weighing, essentially, that you talked about. Here, this shows that weighing doesn't always work, or let the data tell, clearly went wrong here. And here are some other issues, cellular component, where all methods, but I believe this, ultimately my reading here is that the problem is the Go category cellular component. And here, this is human phenotype, so disease-related is somehow maybe a similar problem. We still haven't figured out how to do this right. We hope to get that right. They hope to get that right in CAFA 3. But for molecular function, clearly the scoring is now getting in the right direction. Now the question is, how can we really do a much better effort, or can we improve much over this simple BLAST? What I can assure you is that these top methods have used an amazing amount of information. They use entire networks, they use pathways, they throw anything that you can possibly mine at the problem, excluding text. And it's actually remarkable how little improvement there is. The question is how to push this envelope further, and I believe that remains unclear. I began trying to say something. Why nobody can tell me what I was about to say, right? So let's just